Chapter 13, the spinal cord and spinal nerves. Let's start this chapter by discussing the functions of the spinal cord. The first function is to process reflexes. The second is to integrate excitatory postsynaptic potentials and inhibitory postsynaptic potentials. The third is going to be to conduct sensory impulses to the brain and motor impulses to the effectors. So before we look at the actual components of the spinal cord and the anatomy of the spinal cord, we have to talk about the way we protect it. So it's protected by three major components. Number one, bone. We have all those vertebrae, the ones that we covered when we did the human skeleton way back in our previous lectures. The second protection that we get is from connective tissue. Now these connective tissues are called meninges, and a little later on in this lecture we're going to discuss those three different connective tissue layers and the associated spaces. Now, in one of those spaces, we find this cerebral spinal fluid, the fluid that's going to help cushion and nourish that spinal cord. Let's work off those connective tissue layers, those meninges. Now, we can take them from superficial to deep. So the one that's going to be, we're going to start at number 3, the one that is going to be physically fused to the spinal cord right here is the pia mater. Now, that pia mater is going to have extensions. Those extensions are going to be called denticulate ligaments. Now, these denticulate ligaments are going to attach to the vertebral wall within that vertebral foramen, kind of giving that spinal cord support, holding it in place so that when you have rotational forces on your body, they're not smashing against the walls of that bone. Next is the arachnoid mater. Now that arachnoid mater is going to have extensions as well. Those extensions are going to help keep open the space below it, the subarachnoid space, where we're going to find the CSF or cerebral spinal fluid going. Now the superficial most layer is the dura mater. Now that we've covered the connective tissues, we have to talk about the spaces. Now there are 3 spaces associated with these connective tissue. The first is going to be the epidural space. Now, the epidural space, as the name implies, lies above the dura mater. This is going to be a highly vascularized space. This is typically where we put a saddle block. So if you've ever heard of an epidural, when you're getting ready to deliver a baby, they insert a catheter into this space to kind of numb everything below L2, so that second lumbar vertebra down. Again, typically clinically, it's called a saddle block. Now, the second space is known as the subdural space. So it's the space below the dura mater. The last space is the subarachnoid space. Now, this space is where we're going to find the CSF, the cerebral spinal fluid, flowing around the spinal cord. Now that we have covered the protection of the spinal cord, let's look at the anatomy. We're going to look at this 2 ways. We're going to start by looking at the longitudinal section of the anatomy, so where it begins, where it terminates, and some of those major structures. And lastly, the cross-sectional anatomy, again, of the spinal cord, looking at some specific structures. Now, what does this picture tell us? So this picture tells us that the spinal cord begins as an extension of the medulla oblongata, so way up here at the brain. And it's going to travel through the vertebral foramen of those vertebrae and terminate all the way at L2, which is down here. Now, let's look at a couple things about the spinal cord. We get 2 enlargements, the cervical enlargement here and the lumbar enlargement here. Now, what are these enlargements? Notice that they're going to feed a couple of branches, networks, plexuses. So the cervical enlargement lies by this cervical plexus and the brachial plexus. So we have a lot of sensory information and motor information coming in and out respectively to the spinal cord. So we see an increase in gray matter. So because that increase in gray matter, the increase in the integration center gives us these enlargements. Notice there is no thoracic enlargement because we're feeding here the nerves through here, the nerves of the rib cage, the lungs, etc. So we do not need to have an increase in gray matter or a plexus. Now the lumbar enlargement is going to see that we feed our lumbar plexus and then lower below our sacral plexus. Again, more information coming in from the lower limbs and lower regions, which gives us an increase in the amount of gray matter, which gives us the second enlargement. Now the next question is this termination, the spinal cord terminating around L1, L2. Now the question I always ask my students is, when does the spinal cord stop growing? So if you think about that, think about answering that. When does the spinal cord stop growing? Now, a lot of you have guessed or probably are thinking that it's adulthood or when puberty ends around the age of 20, 24, when in actuality, our spinal cord stops growing about the age of 4. Yes, 4 years old. So the next question people always ask, well, if my spinal cord is done growing at 4, how does it get bigger as I get bigger, right? Well, the answer is pretty simple. It's stretches. So that's it. It stretches. But what's all this other stuff down here below the level of L1 and L2? Well, these are the teasing. I call it teasing of the spinal cord. All these nerves, we get a tail. So the entire tail looks like a horse's tail. So in Latin, they called it cauda for tail, equina for horse. So that's our cauda equina. Now, look at the spinal cord where it terminates. Where before it leads into this tail, it is this cone. So we call it conus medullaris. That's really where the spinal cord ends. Now, from that tail, we're going to enter and we leave all those holes of foramen of the sacra, so sacral foramina that we talked about when we did that bone. And then we're going to end at a little phylum called the phylum terminale, which is going to lead to a coccygeal ligament that is going to attach to the coccyx. So that is the longitudinal anatomy of our spinal cord. And again, in lab, we will cover the plexuses, identifying the plexuses and some of the major nerves that come off those plexuses. Since we just finished the longitudinal anatomy, next is cross-sectional anatomy. So we're going to start on the internal aspect of the spinal cord. So a couple things to notice. We have gray matter and we have white matter. So the gray matter is this beautiful butterfly-shaped structure here, whereas the white matter is everything else around it. So let's go ahead and just delete this and discuss how we name this. Now, the white matter is going to be arranged in columns. So we're going to basically identify the front and back of the spinal cord and then name those columns and subsequently the horns of the gray matter. So we look to this deep slit right here, and it is called the anterior median fissure. Remember, from our bone terminology, we have a deep slit as a fissure. So this is going to be our front because it is anterior. Well, if there's an anterior portion, there's going to be a posterior, but it's not as deep of a slit. It's more of a shallow groove, so we're going to call it the posterior median sulcus. Now that we know front and back, anterior and posterior, we can now name the columns. So we break it up into 3 regions, 1, 2, 3. So we start with the posterior white column, 1, so we'll name that 1. We have the lateral white column, we'll say it's here as 2, this is 2. And then we have the anterior white column, we'll name that 3, okay? That brings us to our horns. We have 1, 2, 3 portions. The posterior gray horn, again, in back. We have, put that as 1. We have the lateral gray horn as 2. And we have the anterior gray horn as 3. Now, a couple things to talk about now is this hole in the center of the spinal cord. It is called the central canal. So spinal fluid, cerebral spinal fluid flows around the spinal cord in that subarachnoid space and through the spinal cord through the central canal. Now, we have a bridge here. This bridge is called the great commissure. Now, the great commissure has a anterior and posterior component, so we have our anterior and posterior. It just follows with the direction of the spinal cord. Now, with the white portion, again, we have an anterior white commissure, so it is this region here, and a posterior white commissure, which is that region there. That brings us to these little rootlets that are coming in and out of the spinal cord. Now, remember, sensory information comes in. We process through a bulge, a knot. So this is called our dorsal ganglia, so DRG, dorsal root ganglia. So this pathway, this is our dorsal root. It is an afferent pathway of sensory information coming in. And we see that sensory neuron synapsing right here. We'll name this number 2 with our interneuron that is taking that signal and translating it and putting it into a motor signal, number 3. Now we move out a ventral root, right? So this is our ventral root. And the pathway is an efferent with an E, efferent pathway exiting. We looked at a drawing of the spinal cord. This is a actual transverse section of the spinal cord, and again the anatomy stays the same. You have the posterior median sulcus in back, the anterior median fissure in front, the central canal in the middle, gray commissure, all the white posterior, lateral, and anterior white columns, and the same with the gray horns, posterior, lateral, and anterior gray horn. So anatomy is exactly the same. One last thing to point out, since we have this real-life spinal cord section up, is the difference between gray and white matter. Now, we introduced this before, but we're just going to reiterate it here. The white matter is white because the axons are myelinated, right? So the myelinated axons gives us the white matter. The gray matter, that beautiful butterfly in the center, is unmyelinated or unmyelinated axons. That's why it presents as gray. So as we discussed on the previous slide, we can use the internal anatomy of the spinal cord to allow for sensory and motor information to be processed in a specific organized way. So what we can see is a couple things going on here. Number 1, we have a picture of our skin. We see that the sensory receptors in the skin detect a stimulus. And again, could be pain, could be hot, could be cold, could be touch, light touch, crude touch, pressure, etc. It sends that information into the dorsal root of the spinal cord, at which point, again, we see these unipolar sensory neurons, and it will synapse here inside of the gray matter where our interneuron is. So again, number 1, we detect the sensory stimulus. 2, we process information through the dorsal root and afferent pathway. Into 3, the interneuron. Now here, sensory information could do a couple things. It's going to ascend, so we get our ascending sensory tracts ascending up the spinal cord to the brain, at which point it can also cross over. This is a contralateral fashion for ascending tract, so this portion A will be ipsilateral, B would be contralateral, and then once the cerebral cortex integrates that, it comes down these descending pathways via a motor neuron, which point it connect in here into the central nervous system, and that's gray matter, a somatic motor neuron, multipolar motor neuron, that moves out. So that would put us at step number 4. So the ventral root processes motor information via that motor neuron. It crosses, so we go here because we didn't really talk about this on the other picture. This is our spinal nerve, so our spinal nerve where the dorsal and ventral root meet, and the first place that our sensory and motor neurons intermingle. And then we follow out to step 5, which is our effector, so it goes to the skeletal muscle. That brings us to those spinal nerves that we just got finished circling, and we can count them. So spinal nerves connect the central nervous system to sensory receptors, muscles, and glands, and are part of the PNS, or the peripheral nervous system. We have 31 pairs of spinal nerves. Anterior and posterior roots attach to the spinal nerve to a segment of the spinal cord. So again, we go from those rootlets to the spinal nerve, which will extend out to the central nervous system. Now, counting spinal nerves is a little bit tricky. If you think about the vertebra that we have, so we have 7 cervical vertebra, 12 thoracic, 5 lumbar. When we count spinal nerves, we count either above or below the vertebra. Now, if we were to start at our cervical spinal nerve 1, we're going to count the spinal nerve above C1. And then we have CSN2, which is above C2. And we continue all the way down until we get to CSN8. Now we say, well, wait a minute. Why do we have 8 cervical spinal nerves when we only have 7 cervical vertebra? Well, again, we're counting the space above. right? We're counting the space above. So we have to account for that space between the occipital bone and C1. There's going to be a spinal nerve there, etc., etc. Now we start at T1. Now, instead of counting above, we now count below. So below T1 is TSN1, then we have T2, and that gives us TSN2. And we go all the way to, and we're going to carry up here, we go all the way to T12, which gives us TSN12. And then we have L1, LSN1, L2, all the way down to LSN5, which is above L5. So for cervical spinal nerves, we have 8, so 1 more than our vertebra. For thoracic spinal nerves, we have 12 that aligns below each of our 12 vertebra, and 5 lumbar spinal nerves below each of those lumbar vertebra. And that ultimately leaves us with 5 sacral nerves, spinal nerves, and 1 coccygeal spinal nerve. Total 31 pairs, so we have 1 on each side, of spinal nerves. Connective tissue coverings of spinal nerves. So similar to muscles, we're going to see a similar nomenclature and very similar arrangement to our spinal nerves, to our nerves that we saw again with our muscles. So let's start with our analogy of those bundles within a bundles. We'll start big. We'll take one entire spinal nerve. We're going to cover it with a connective tissue, and it will be called the epineurium. Now, we're going to bundle more of these nerves, and those bundles are going to be called fascicles. So let's pull a fascicle out. We're going to wrap that fascicle, that bundle, in a connective tissue, and it's going to be called the perineurium. Now we're going to take an axon. Okay, so one single neuron. And we take that neuron, we see that it has this myelin sheath wrapping, and we're going to cover that neuron with another connective tissue sheath, and that is our endoneurium. So again, from outside in, superficial to deep, epineurium, perineurium, and our third is our endoneurium. So again, we have a bundle of bundles. Now, some differences to muscle. We don't have bundles within bundles within bundles, but this is quite simply arranged like those similar bundles. And again, the nomenclature, instead of epimysium that we saw in muscle, we see epineurium, that neuro for nerve. So similar nomenclature. Now, if we look at an actual transverse section of that spinal nerve, we see that centrally localized blood vessel, right? And then we have a bundle, a bundle, a bundle, another bundle. So all these fascicles of axons, and you can actually see, we're going to zoom on in real quick, you can see that myelin sheath, and then the black in the center is where the axon would move through. Branches of spinal nerves. So now, shortly after passing through its intervertebral foramen, a spinal nerve divides into several branches known as rami. So if we look at one rami, we have these rami communicantes. So here's again our spinal nerve. We get this branch that leads to a sympathetic trunk chain ganglia. So this is part of our autonomic nervous system that's going to move this fight or flight information up and down the spinal cord following a chain, following these ganglia. From there, we're going to see an anterior ramus, right, also known as the ventral ramus. So we see this root carrying this way. We're also going to have a posterior or dorsal ramus, so again, off another branch coming off into those deep back muscles. So the largest of these roots is going to be that anterior ramus. And then what we can do here is actually look at these spaces and layers of the meninges that we didn't really go through before. So let's look at some of the layers, and we're going to start deep, right? We're going to start the layer that's fused right here against the spinal cord, that pia mater. And you can see, look at that white denticulate ligament, that extension of the pia mater. From there, we find these, if we come up here, we go up here into these little white spaces, those extensions of the arachnoid matter that is going to give us where CSF, so that's our subarachnoid space, our arachnoid matter right above it, and then our dura mater above that, and then this big space here is our epidural space. This is where we talked about those saddle blocks, so that's the space above the dura. And again, if we zoom on in, and the pixelation here makes it kind of hard, but in between the dura and the arachnoid is a space, so that would be our subdural space, quite small. Now on Blackboard, we have a handout for all the spinal plexuses. So again, review your plexuses. You have your cervical plexus. We have our lumbar plexus. We have our sacral plexus. Now for each of these plexuses, you will also on that handout find the associated nerves that branch off and originate from these plexuses, where they go and what they do. So again, please review this handout on Blackboard. It is pertinent information to this lecture that we do not cover in actual lecture. If you do happen to have any questions about this handout, please feel free to reach out. Now, we talked about the spinal nerves, but we didn't really talk about where they go and what they innervate. This gives us a map. They're called dermatomes. Now, certain segments of the skin are supplied by spinal nerves that carry somatic sensory nerve impulses into the spinal cord and the brain. Now, if you look, you see that we have divisions that align with the divisions of the longitudinal section of our spinal cord. Namely, the green is where we find our cervical plexus. Our thoracic region stays intact right about L1, L2, where that second enlargement was. We have our lumbar plexus followed by our sacral plexus down here. Now, what are the clinical significance of these dermatomes? We'll see that doctors and EMTs can pinch certain regions of the body, and based on knowing this mapping of dermatomes, the mapping of this innervation of the peripheral nervous system, they can assess what part of our spinal cord is damaged. So we could put a little more clinical technicality to it. So we can say that basically this nerve root that's supplying all these neighboring dermatomes overlap. So sectioning or dysfunction of a single dorsal root produces this diminished sensation. Now clinically, diminished sensation is known as hypoesthesia. Now, if you have a complete loss, a total loss of sensation, this is known as anesthesia. Now, you can get anesthesia in certain regions that are supplied predominantly by that dermatome, but for dermatomal anesthesia, it requires damage to typically at least 3 dorsal roots. We can also look at another example of where these dermatomes come into play. So let's look at a herniated disc, right? And one of those intervertebral discs herniated. Now this can produce sharp radiating pain within the distribution of the affected dermatome, and you can see this. So if we look at where that pain is felt in through the buttocks down and radiating down the outside anterior lateral aspect of the leg and through the back of the leg, we can feel this because of this dermatomal map. Let's now examine spinal cord physiology. So the question we're going to ask is, how does information travel in and through the spinal cord? Now, remember, the white matter is myelinated, so white matter tracts conduct nerve impulses to and from the brain. Those unmyelinated gray tracts, those gray matter, receives and integrates incoming and outgoing information to perform spinal reflexes. So let's look at some of these tracts. So let's look at some sensory and motor tracts. Now, realize that anything blue in this picture is going to be sensory, anything red is going to be motor. Now, we are going to go into what these tracts specifically do, but I'm going to give you a little cheat to help you identify sensory and motor. So let's look at some of our motor. We have our lateral cortical spinal. We have a rubrospinal, a lateral reticulospinal, a vestibulospinal, and we have another medial reticulospinal tract. Now, for sensory, we have the gracilis fasciculus, the cuneate fasciculus. These are posterior columns. We have a posterior spinocerebellar, anterior spinocerebellar, spinothalamic. Now, again, realize that even though this picture kind of divides it in half with blue on the right and red on the left, remember that we get both sensory and motor overlapping. So this space right here would be reserved for the lateral cortical spinal and rubrospinal tracts that would fit into this little section right here. And again, if you look at this section hugging here on the left side, clinically or aspect-wise would really be the right, but from our perspective, on the left side would be that spinothalamic. Now, what is the cheat? There is a cheat here, and this will help you get through a quick identification, sensory versus motor. Now, here it is. So I'm going to highlight in red for motor. Now, anything that says spinal, see that word spinal? Spinal, 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 spinal. That is our clue for motor tracks. Anything that says spino, so spino, 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 that is our clue for sensory tracks. So there's your quick and dirty way of identifying sensory and motor tracks. Now again, we are going to get a little more specific and identify specifically what they do and where they go. But just for the real quick and dirty, if it's motor or sensory, that's your little trick. Spino for sensory, spinal for motor. Let's talk about reflexes and reflex arcs. Now, a reflex is fast, it's predictable, it's an autonomic response to changes in the environment. So reflexes help to maintain balance or homeostasis. The spinal cord serves as the integrating center for our spinal reflexes. We are going to put this to a 5-step process. And we're going to see that integration takes place in the gray matter of the spinal cord. Now, if we look at quick examples before we get into much detail, think about touching a hot stove. Now, when you touch a hot stove, what happens? You touch that, your brain says, your skin says, something hot, this could be dangerous. It sends that signal to your brain. Your brain says, I have to remove my hand. I don't want to burn it permanently from that hot, dangerous situation. And it causes a response, a withdrawal, a flexor response, moving your hand away from that hot stove. Now we think about that as being really fast. So this is our fast, predictable response. We say, how fast is it going? Well, it can vary from reflex to reflex, but typically these are going anywhere from 150 to 300 miles per hour. That's how fast these signals are moving, and you think about time, it takes about 0.15 seconds to complete an entire cycle from recognition of that stimulus all the way to the effect, the response. Let's put it to steps, and we've outlined these steps before, but now we're going to simplify it a little bit. So again, we're outlining the general components of a reflex arc. So step number 1, a sensory receptor in our skin is going to detect and respond to a stimulus that's producing a generator or receptor potential. That stimulus is going to travel down that sensory neuron into the dorsal root, through that dorsal root ganglion. So step 2 will be our sensory neuron. Axon is going to conduct impulses from the receptor to the integration center. So we're now deep in the CNS, in that central nervous system, where we find step 3, our interneuron. So the interneuron is our integrating center. So this is where one or more regions within the central nervous system is going to relay impulses from sensory to motor. This is the go-between. This is the translator. It is taking that information coming in and deciding how to respond to it. Well, that gives us step 4, our efferent or efferent pathway via our ventral root. We have that multipolar motor neuron moving out. So axon is going to conduct impulses from the integration center in the CNS all the way to the peripheral nervous system out to the effector. Now that effector can be a muscle or gland that's going to respond to that impulse. So when we used our example of the hot stove, we are going to talk about varying types of reflexes, simple, monosynaptic, polysynaptic, etc. We are going to talk about how this happens. Again, think about that hot stove. You touch that hot stove, that's step 1. Information travels 150, 300 miles per hour into the central nervous system. The brain says, ow, that's hot, remove my hand. And then the signal moves out to the skeletal muscles in our hands, in our forearms, in our bicep and tricep, and causes a flexor reflex. So our biceps react, biceps flexes, pulls our hand away from that hot stove. Now let's throw some more terminology in here. Remember we talked about ipsilateral and contralateral, and we said that we were going to reserve contralateral. We're going to reserve these terminologies for the reflexes. So again, ipsilateral is same side, whereas contralateral is opposite side. So let's look at this reflex. And let's say if this is an ipsilateral or contralateral. So look, stimulus comes in on one side, it processes and travels through the same side. So this reflex happens to be an ipsilateral. If it was a contralateral, we see information moving across that great commissure to the other side, to the right side of the spinal cord. This would make it contralateral. The next terminology we're going to throw in here is monosynaptic and polysynaptic. So take a look at this picture and see if you can guess which one it is before I give you the answer. So take a look, try to count the synapses. So let's have a look. We count sensory, synapse 1, interneuron to motor, synapse 2. So this is more than one. So this reflex here, the simple reflex, is a polysynaptic reflex. So then you're probably saying, well, how do I get a motor reflex? Not a motor, but a monosynaptic reflex. Well, the answer is pretty simple. Who do you have to get rid of? Get rid of the interneuron. By getting rid of the interneuron, we put sensory direct to motor. These are simple stretch reflexes, and this gives us a monosynaptic reflex. And we are going to elaborate on this in the next few slides. So here's some of our terminology. So first was ipsilateral, so we said that was same side. Next was contralateral, and we said that was opposite side. Then that gives us monosynaptic versus polysynaptic. That's one, and this would polysynaptic would be two or more. Now, reciprocal innervation. Now, this is a little bit different. We have to think about muscle. So think about the prime mover and the antagonist, right? So if you think about muscle groups, this helps us understand reciprocal innervation. So think about that flexor reflex that we just talked about on the hot stove. Now remember, in order for us to withdraw from that hot stove, the bicep has to contract concentrically, when the tricep's going to contract eccentrically. So we're going to get a contraction versus a relaxation. Now that innervation to the bicep, communication to the bicep to contract, and communication to the tricep to essentially relax, is that reciprocal innervation. Next is intersegmental. This is a simple definition where we get input sensory and the motor output nerves are going to be connected by tracts running within the spinal cord that can either move up or down the different segments of the spinal cord. So intersegmental means that we can travel up or down the spinal cord through different segments. So we're accessing different segments. Now, let's look at a couple specific reflexes. The first is a stretch reflex, and you're going to do this in lab. You've had doctors do this to you during physicals all the time. This is a patellar knee jerk. So this is going to help control muscle length by causing muscle contraction. This is also a very simple, fast, monosynaptic reflex. So let's look. We have a stimuli coming in, which is the striking of that patellar ligament. That's going to send a sensory response from the muscle into our spinal cord. So we find sensory, same muscle contracts and relieves the stretching. And again, look, one direct connection from the sensory neuron to the motor neuron, monosynaptic reflex. Now we go to the exact same muscle. So again, that stretching stimulates the sensory receptor, step 1, sensory neuron, integration center number 3, so monosynaptic because we go from sensory right to motor, to that motor neuron pathway to the effector number 5. Now you say, well, what is this dashed line? Because this is a polysynaptic reflex. Well, this is the reciprocal, the inhibitory interneuron, the reciprocal innervation, where we're talking to those hamstring groups, the antagonistic muscle groups, to relax. So we get that quick knee jerks by accessing the quadriceps and relaxing the hamstrings. The next reflex we're going to look at is a tendon reflex. So these are going to help control muscle tension by causing muscle relaxation. So if you look at where the sensory receptor is, it's in a little different place. It's in our tendons that are connecting the quadriceps to that patella. So let's look at the steps. So step 1, increased tension detected by the tendons of the muscles of the quadriceps is going to stimulate a sensory receptor or a tendon organ. We fire that information through that sensory neuron, number 2. Now we get these polysynaptic, look, polysynaptic neurons. And we get two pathways. Another dashed pathway, that's that inhibitory pathway, that reciprocal innervation that's going to talk to the quadriceps to say, hey, relax, we don't want you to contract. There's already too much tension on this tendon. So it's going to cause it to relax. While at the same time, to help relieve that tension, it's going to cause a motor response to the antagonistic muscle groups, the hamstrings, to contract, causing this knee jerk, reverse knee jerk. So we get our heel moving back towards our buttocks. The third reflex we're going to look at is a flexor or withdrawal reflex. Now, this was the example we used with the hot stove. I like that visual a little bit better, but let's take a look at this one. Now, let's say that your foot steps on, say, something sharp like a tack. So stepping on a tack stimulates a sensory receptor, so the pain receptors in our skin, and it moves via our sensory neuron, again, into our interneuron, and then our muscle response, so polysynaptic, to our hamstrings saying contract to give us the withdrawal from the leg. Okay, so now again, notice that this is a ipsilateral polysynaptic reflex. Another scenario would be our that hot stove scenario where you touch the hot stove, so that hot detects in our brain, tells us to move our hand away. That's our effect. The fourth and most complicated one is the crossed-extensor reflex. So remember, this is again a polysynaptic and a contralateral reflex. So let's see what happens. Same scenario, you step on a tack, the information travels up the spinal cord, polysynaptic, goes to the hamstrings to say flexor withdrawal. So we're going to move our leg from that painful stimulus. Now, what's happening here? Why is it a contralateral reflex? So we're going to one leg, right? We're going to the right leg to remove that leg with that flexor response with the hamstrings contracting to move it away from that painful stimulus. But now to protect us so that we just don't fall every time we step on like a sharp rock or a Lego or a tack, we have to activate the muscles of the quadriceps in the opposing leg. So notice we're going across the great commissure to the other side of the body. By going to the other side of the body, we're going to contract the quadriceps, which keeps that left leg extended or straight. And again, this is maintaining balance. If we didn't have this crossed-extensor reflex, every time you stepped on a Lego or a stone or a tack, you'd simply fall to the ground. Now, that wouldn't be too conducive to our health, so thank God we have these contralateral reflexes. Now, let's look at some clinical connections. Why do your doctors do these reflexes? Why do they hit your knee with those reflex hammers, or your inside of your elbow, or the back of your calcaneal tendon? Now, these reflexes are often used for diagnosing disorders of the nervous system and locating injured tissues. So, for example, when they shine a light in your eye, you should see a pupillary light reflex where the pupil constricts down, and then when you remove that stimulus, the light, the pupil dilates. Now, if a reflex is absent or abnormal, the damage may be somewhere along a particular conduction pathway. Now, we also look at babies. As soon as a baby is born, they go through this Apgar test where they wisp the cheek and the baby should suckle towards that stimulus. They touch the palm, the baby should grip and clasp their hands. And then the last one that we do in lab is called the Babinski reflex, where they make almost like this Nike swoosh from the heel to the big toe, and you will see a specific response. So like dorsiflexion and fanning of the toes in a baby is an abnormal response. It could signal neurological dysfunction. Now again, for homeostatic imbalances, damage that results from traumatic injuries depend on a couple things. Number one, depends on the degree of spinal cord section, so where that damage occurred, or the degree of compression of the segments involved. So again, we talk about things like paralysis or loss of feeling, where when you have traumatic injury like a car injury, people could be temporarily paralyzed to certain regions of their body because of massive damage and swelling. But again, things like that, we talk about degree of a compression or degree of injury to a section. We say that with time, when that swelling minimizes and goes down, we usually regain movement or feeling in that area. Now with severing spinal cords, again, depending on where we sever the spinal cord, we know we could, these types of injuries cannot be repaired, and we understand that. We talk about words, clinical words like quadriplegic, paraplegic, and again, this is dependent on the level of the spinal cord that has been damaged or severed.